Howdy, this is Mr. Justice. Today we're going to do some more draw notes, answering the question at the top of this page. How does each step of cellular respiration contribute to the total oxidation, the breakdown of glucose to ATP? Now, you can make a copy of this if you have a printer at home. You can find this on Canvas under day 37 for the draw notes for aerobic respiration. I've also given you guys a scan of this and put it into a Google Drawing. So if you don't have a printer to make your own version of this page of notes, then you can still participate by creating the arrows and the different stuff that we're about to do here by doing that through Google Draw. And by the end of the process, we're going to have something that looks just like this, all right? I suggest that you grab some color pencils if you have them, markers, different colored pens. If you don't have that, just a blue pen will do. Or a pencil, I suppose. But if you want to make it look nice and pretty like this one, then I suggest getting some colors. All right, so let's get started here. I'll move this out of the way, and we're going to talk about the total oxidation breakdown of glucose to ATP. All right, so I'm going to use 6 different colors, and these colors will all kind of represent something. For example, all of my arrows that I draw today will be blue. Do that right there. So the very first step in the total oxidation of glucose to ATP is our very first step here. It actually happens outside of the mitochondria. This process is called glycolysis. Glycolysis is when glucose, a 6-carbon sugar, is split into 2 3-carbon pyruvates. There's 2 of these here. This actually takes a little bit of an investment of energy to get this going. 2 ATP are required. So I'm going to use red for me to kind of symbolize the inputs, the reactants required for the process to move forward. We've got to incorporate use 2 ATP to make 4. Right? I'm going to use a different color to represent the outputs, the products that are made here. We make 4 ATP. Hey, it took 2 ATP to make 4, so we say that this process nets 2 ATP. There's a net gain of 2 ATP molecules. ATP is used by our cells to do work. It's a usable form of chemical energy here. Glucose not so usable, the ATP is, but we only make 2 ATP during glycolysis. Now, we've talked about this before. This pyruvate, if no O2 is available, no oxygen is available in the cells, then this pyruvate will be oxidized through fermentation, and well, we make some products, right? Alcohol fermentation will produce alcohol and carbon dioxide. Alcohol fermentation is performed by yeast. And then there's also another type of fermentation called lactic acid fermentation that bacteria, animal cells, and even our muscle cells undergo. Lactic acid fermentation occurs when no O2 is available. You guys have felt this before. If you ever really exerted yourself, maybe you're running in a marathon, and you start to feel a pain that starts to accumulate in your muscles, that's the lactic acid that is being produced by glycolysis and lactic acid fermentation. The reason why that happens is because our muscle cells are working so fast, it's zooming through the oxygen that we're breathing in, and it's not getting enough. Some of the muscle cells aren't getting enough of that oxygen gas to allow, well, the total oxidation of breakdown of glucose, which we'll get to in a moment. And so instead, there's another metabolic pathway that allows us to continue to make some ATP so that our muscles can continue to do work. If you want that burning to go away, that lactic acid buildup that you might feel when you're really exerting yourself, all you have to do is slow down. Take some deep breaths again. Get that oxygen flowing. Oxygen's present, so we're going to allow this pyruvate then to migrate into the mitochondria, and that's where we are here. Allow me for a moment here. I'm going to go through a couple other processes here. I'd like to mention where these processes are occurring. So the cytoplasm is outside of the mitochondria. This is the area of the cytoplasm. This is the jelly-like fluid that exists within the cells. It contains dissolved solids, or dissolved salts rather, and enzymes. in the cytoplasm. And then take a look here. We've got on the other side of these two lines here is the inside, the very interior of the mitochondria. This is the mitochondrial matrix, we'll call it. So pyruvate moves into this area here. It passes through two membranes. The mitochondria is made up of two membranes, the outer membrane and the inner membrane. So these two membranes here separate the mitochondrial matrix from the cytoplasm. And then take a look, there's another area that's important to us that we'll learn a little bit more about in a moment. Between the outer membrane and the inner membrane, this is the inner membrane space, and that will become important when we start really making a lot more ATP in a moment. All right, so a couple things here that I'd like to point out as well. Take a look. The NADH, I'm going to put two little dots on the top of the NADH. And I have this little information here about NADH. Allow me to read it. The NADH made during glycolysis will be used in fermentation if no O2 is present, or in the electron transport chain if O2 is present. So this NADH, this high energy electron carriers, is carrying electrons. Some of them were involved or incorporated with glucose. They're now being held here. This chemical form of energy will be used later on in the process here if oxygen's present, all right? And take a look here, right? You saw me put these little dots on top of NADH. Let's do that again here and label them so we know that these two dots represent the electrons being carried by NADH. All right, lots of information so far. All we've talked about is glycolysis. We split glucose into pyruvate. Pyruvate, if oxygen's present, will move into the mitochondria. And real quick, just a quick modification of pyruvate into another organic compound called acetyl-CoA. And acetyl-CoA, let's think about this. We've gone from a 3-carbon organic compound to a 2-carbon organic compound. Where did the carbon go? Well, carbon dioxide was removed as a waste product as we transform 3-carbon pyruvate into a 2-carbon acetyl-CoA. Since this is a product here, I'm going to go ahead and highlight it using my color that I'm saying represents products, what comes out of the situation. Well, carbon dioxide. Let me make a real quick connection with you guys about the respiration dance that we learned before, right? So let's just take a moment and think about this. Let's sing it together, in fact. Ready? The sun shines down, electrons are excited, energy is stored in the bonds of glucose. Animals eat it, digest it, transport it. Lub-dub, lub-dub. Glucose enters the cell with the help of insulin. It's broken down into pyruvic acids, which enters the Krebs cycle. There's more to this song. Give me a moment. Think about this. Glucose enters the cell with the help of insulin. I've got 6 fingers up because my fingers represent the carbons in that C6H12O6 glucose. During glycolysis, we break that 6-carbon sugar into 2 3-carbon pyruvates. And then notice what I do which enters the Krebs cycle. I go from 3 to 2 fingers. That's supposed to represent the 3-carbon pyruvate modified into 2-carbon acetyl-CoA. So we go from 3 to 2. Acetyl-CoA is actually an enzyme. It's holding those 2 carbons. And I wanted to point this out because it's not 100% obvious. Like the carbon just leaves the system. How is that actually going down? I've got a little note up here. So these processes are enzyme catalyzed. Now there's a good $2 word for us. Enzyme catalyzed. What does that mean? This means that enzymes are helping the reactions happen. And so even though they're not mentioned, the enzymes associated with these processes aren't mentioned on our page of notes. They are there. They are what is multiple enzymes, in fact, are splitting glucose into pyruvate. And another enzyme is going to modify pyruvate to acetyl-CoA. So I just wanted to point that out, that enzymes are helping this process happen. Enzymes are a class of protein. All right, so let's grab our color for our arrows. If you are following along on the Google Draw version of this, note that you can use any of the shapes to do, and the line tool to do all of this. So I've taken the acetyl-CoA and I've suggested that it enters into the Krebs cycle where electrons are stripped off. Additional enzymes are making the Krebs cycle occur. These enzymes are ripping apart, stripping off those electrons from those organic compounds, and well, the carbon moves out of the system, right? I'm going to grab my color to represent products. The carbon dioxide is being produced here and here during this process. That carbon dioxide is exiting our bodies. As humans, we're doing this process ourselves. So we also make a little bit of ATP. Allow me to underline this. And then, you know, electrons are stripped off. Where are they being held? The electrons are being held by this NADH and FADH2, another form of chemical energy here. So let me just attach those cute little electrons to those two chemicals there. Yeah. All right. Oh, and you know what? Since we're making ATP, let's go ahead and draw that nice star around this so that we can easily see that a little bit of ATP is being produced during the Krebs cycle. Let's go back to that song now. Glucose enters a cell with the help of insulin. It's in the cytoplasm. It's then broken down into 2 pyruvic acids. That happens here with glycolysis. Oxygen's present, which enters the Krebs cycle. And then remember, with our song, what we do is carbon dioxide is exiting the system, and notice what I'm doing with my fingers. I'm going from 2 to 1 to 0. All of the carbon is now exiting in the form of carbon. The rest of the carbon is exiting the system in the form of carbon dioxide. And those electrons that were holding the organic compounds that used to be glucose, that used to be pyruvate, that used to be acetyl-CoA, those electrons are now being held by that energy, NADH and FADH2. I put this here for us because I wanted us to have that connection with that respiration song that we learned about earlier in the class. And so I'm just going to add a couple cute little notes here to help us remind ourselves, yeah, this is a song. How about a little treble clef? Look at this. I think I'm doing that right. Yeah, there we go. All right, there's a little song for us to remember. Electrons are stripped off, and if you remember, we go from electrons are stripped off and then go down the electron transport chain. So our 2 last fingers on our hands during that song represent the electrons being held by NADH and FADH2. Now take a look. These electrons are passed to additional enzymes, so these proteins that are embedded within the inner membrane of the mitochondria. That's why I've kind of written the words electron transport chain proteins within this line. So take a look here. Allow me to grab my color to represent my lines. So the NADH and FADH2 hand off or pass off the electrons to the different proteins here in the electron transport chain. And each of these proteins will hand one another the electrons, and boop. At the end of the electron transport chain is oxygen. So let me use my color that represents the inputs. Oxygen is required. We remember that from earlier in the lecture. Oxygen, why? Oxygen is really good at accepting electrons and taking electrons. It's a good oxidizer. That's what oxidizers do, is take electrons. Oxygen's really good at it. It's got high electronegativity. And so at the very end of the electron transport chain, if oxygen's present, it will grab these electrons. And when it does that, it reacts with another H+ ion that might be present, and we produce a little bit of water. So water is being made, right? Take a look. This is the other major output here from this process. Oxygen, electrons plus an H will give us H2O. So we're making water. Now, if you don't believe me, right? We're making water? What? As humans, we make water too. It happens when oxygen's present. Oxygen accepts the electrons at the end of the electron transport chain. We produce water. Where is that water then? We're blowing out carbon dioxide. We're also blowing out water vapor too. go into the bathroom, get close to the mirror, and blow on. I wonder if I can do this on the, maybe, I don't know. That condensation is the water being produced here. Take a look, guys. There's different chemicals, like cyanide is a poison. And what cyanide does is it doesn't allow oxygen to accept the electrons at the end of the electron transport chain. So if there's not something, oxygen, that can accept the electrons, then the electrons at the start of the electron transport chain won't continue to move down. And because of that, then everything grinds to a halt. Fermentation occurs in our muscle cells, but that might not be enough ATP to make all of the rest of our faculties continue to function. That cyanide is a poison because it will kill you. It stops allowing oxygen to accept the electrons. And so we stop making the bulk of our ATP. Now, what are these electron transport chain proteins doing? Let me grab a different color. What these guys do is they use the energy in these electrons to pump H+ ions into the inner membrane space. So I'm going to draw a couple H+ ions, and I'm going to draw a bunch of them here because I want us to know that the concentration on the inside of the inner membrane space has a higher concentration of H+ ions than in the matrix. Let me put one H+ right over this guy right here. Okay, we'll get to that in a moment. I wanted to point this out here. So in nature, just like think of blowing up a balloon. If you blow up a balloon, the pressure on the inside of the balloon is higher than the pressure on the outside. And so when you let that balloon, the opening of the balloon open up, that air will rush from an area of high pressure to an area of low pressure. Well, something similar is happening here with these H+ ions that are accumulating in a high concentration on the inside of this inner membrane space. This high concentration, these H+ ions are charged. They're unable to pass across any one of these membranes. So it's kind of like we're blowing up a balloon. And there's a tendency for these H+ ions to want to move from an area of high concentration to an area of low concentration inside the matrix. Now, they're not able to pass through that membrane. The only way that they're able to move back into the matrix is via or through this ATP synthase protein here embedded within the inner membrane. So let's just draw that arrow of the H+ moving through ATP synthase. And what ATP synthase is able to do, it's a protein that is able to harness the kinetic energy, the movement of the H+ ions, and then attach a phosphate group to create ATP. In fact, we're making the big bulk of our ATP is being produced here during this process. Upwards of 32 to 34 ATP molecules as a result. Let me just color this in here. Let me color in my ATP synthase protein as well. Yeah, we're making the bulk of the ATP here, right? Take a look. Let's add it all up. 34 plus 2 plus 2 is upwards of 38 ATP from one molecule of glucose. That answers that question. How does each step of cellular respiration contribute to the total oxidation breakdown of glucose to ATP? Glycolysis splits glucose into pyruvate and nets to ATP. If oxygen's present, pyruvate is modified into acetyl-CoA, which is further stripped of its electrons during the Krebs cycle. We produce a little bit of ATP, some carbon dioxide, and those electrons from those organic compounds here in Krebs cycle are being held by NADH and FADH2. These two high-energy electron carriers will pass their electrons to the proteins embedded in the inner membrane. These proteins will allow the electrons to move down the electron transport chain. Oxygen accepts the electrons at the end of the electron transport chain to produce water. These electron transport chain proteins use the energy from these electrons to pump H+ ions into the inner membrane space. The inner membrane space has a higher concentration of H+ than on the in the matrix. These H+ ions will move from an area of high concentration to an area of low concentration through ATP synthase. And when that happens, we produce upwards of 34 ATP. Real quick, last thing that I'll say before we finish our lecture today is it's important to note that the ATP, the NADH, and the FADH2 that our cells are using can actually be reused and recharged. You actually go through about your body's weight in ATP every single day. So that means that you're not eating, 150 pounds or so of food. You're just reusing these molecules. This ATP can, once it's used, it goes from ATP to ADP, and then we can attach another phosphate group here during this process to recharge it. And that third ATP, that third phosphate group in ATP has a lot of potential energy to do some work for us. So I hope this was helpful, and let me know if you have any questions.